Hello, everyone. This is Dave Iverson, the contributing editor at the Michael J. Fox Foundation and the moderator for today's Third Thursday webinar. As many of you know, you can log into these Third Thursday webinars at 12 o'clock Eastern every third Thursday of the month and join us as we take up a topic that's important to our Parkinson's community. We hope you'll continue to do that throughout the year. Our topic today is one of particular importance because we're going to get an update and an assessment of where we are with finding a way to slow down or stop Parkinson's disease. We've made lots of progress in coming up with improved symptomatic treatments, but of course the end goal is to figure out a way to actually stop the disease itself, and that's going to be our focus today. Along those lines, you can submit questions throughout the hour. We'll be keeping track of those, and then starting in about a half an hour, we'll start taking your questions. So you'll see the little Q&A box on your screen, and you can type your questions in there, and then we'll get to as many of those as we can. And as always, we won't be able to get to every single one, and so we want to give you kind of a bonus opportunity to take this topic up further, and that'll be tomorrow on Twitter at 12 noon. The hashtag is FoxPDChat, and we'll continue our conversation tomorrow on this same subject. Two other quick last points, and then we'll get started. You can download the slides from today's webinar. There's a box where it says resource list, and you can click there, and the document will open in a different browser, and you'll be able to either download, save them, print them, whatever you'd like to do. And last but surely not least, we want to thank our sponsors for the Third Thursday webinar series, which are Acadia Pharmaceuticals, Medtronic, and Teva. So thanks to them in particular. All right, let's get started with our topic today. And here are some of the things that we're going to be trying to cover in our conversation today. We're going to take a look at what are the various things that we can do to actually hopefully bring about a change in stopping disease progression. So we'll be taking a look at a variety of things, and we seem to be having a bit of trouble with the slide coming up, at least on my screen. So I'm just going to read you some of what we're going to be trying to cover today. We're going to take a look at the varied approaches to stopping Parkinson's disease. We're going to look at how our immune system is actually able to fight Parkinson's disease. We're going to take a look at what we've learned so far from being able to observe certain compounds that may either slow down the disease itself or reduce disease risk. We'll talk some about something called neurotrophic factors, and then last we'll talk about the things we have to do in order to be able to test these various therapies and actually learn whether or not they're going to work. So let's meet our panelists and find out who it is that's going to be joining us in our conversation today. So here's who our panelists are today. And again, I'm sorry that we're seeming to have some trouble with our slides, but there we go. We've come up with this one. So let's meet our panelists. Bill Sheppard joins us. He is the Chief Financial Officer of Global Wealth and Investment Management at the Bank of America. He's also part of the Michael J. Fox Foundation Leadership Council. Bill was diagnosed with Parkinson's disease in 2006. Bill, thanks for being part of our discussion today.
Thank you for having me, Dave.
Happy to have you join us. Joining us as well is Dr. Lorraine Kalia. She is a neurologist at Toronto Western Hospital and also an assistant professor there at the University of Toronto. Lorraine Kalia, thanks for being part of our discussion as well.
Thanks for inviting me. I'm glad to be part.
And last, Todd Scher joins us. Dr. Todd Scher is the CEO of the Michael J. Fox Foundation and also spent some time as a research scientist before joining the foundation, led those research efforts before he became the CEO of the foundation. Todd, thanks for being here too.
Thank you, Dave.
So we're going to begin our conversation now by taking a look at the various ways that we can try to stop Parkinson's disease. And you see on your screen three different ways of outlining those approaches. Todd, why don't you start us out by just describing the main culprit that we think of these days, and that's the efforts to break up the sticky protein alpha-synuclein, which we think is so central to leading to the cell death that happens in Parkinson's.
So one of the main characteristics of Parkinson's disease is the presence of these clumps of protein that are made up of the protein alpha-synuclein. And it is believed that these protein clumps play an important and critical role in the loss of cells in the brain that are affected in the disease. So a main area of research is to try to now develop therapies that could remove these clumps from the brain, either to help the brain degrade the clumps or even prevent the clumps from forming in the first place.
And we're going to take a look at some of those strategies in just a moment. And Dr. Lorraine Killia, pick up on the second bullet point that we see on the screen. We've learned these interesting associations in recent years, and we're not sure yet whether they're associations or whether there's something that might actually have a causal effect in terms of being able to either reduce the risk of ever getting Parkinson's or slowing it down if you do have it. Describe some of those interesting things that we've begun to observe and what it is that we're now trying to test.
Yeah, so it's been quite fruitful, one field of investigation called epidemiology, which looks at populations and tries to understand a disease by looking at large numbers of people. And so what epidemiological studies have shown us has been that there are certain things that seem to be associated with either a reduced or an increased risk of Parkinson's disease. It doesn't prove causality, meaning just because we see these associations doesn't necessarily mean what we see as the factor that's associated is actually a cause, but it actually provides us with good hints as to what we should be studying further. So I know that we're going to talk more in detail later on about things like nicotine, certain types of blood pressure medications, and also urate in the blood that have been found when you look at large populations of people with Parkinson's disease that either taking one of these medications or having a higher level of one of these chemicals can actually be associated with a lower risk. And so that has really pushed forward an important area of investigation in terms of looking at some of these drugs and modifiers to be able to make a difference in disease modification for Parkinson's disease.
It is a particularly fascinating area, and we're going to be going through that as well as our last bullet point, which has to do with ways that we could actually protect the brain and nurture the cells that still exist. So let's dig right into some of our specific strategies that we're approaching, particularly with this first area of fighting those sticky proteins known as alpha-synuclein. And Todd Scherer, we see on that first bullet point, has to do with actually using the body's immune system. The analogy that's sometimes made here is that this is a bit like what happens when you get a flu shot, but it's a little hard to get your head around. So describe this a little bit more, and then we're going to talk about some specific approaches that are now being tested. But how is it that the body's own immune system could actually fight something like this sticky protein alpha-synuclein?
So there has been evidence in Alzheimer's disease, in some therapeutic approaches for Alzheimer's, and some early studies in Parkinson's, that the body itself might have the capability to target these protein clumps and degrade them. And if you think about the analogy that you used, Dave, to some vaccines or other immune type of therapies, the protein clump is actually a foreign, unfamiliar body that is exposed to the body that normally wouldn't be there. And what your immune system is actually designed to do is to attack any foreign body or pathogen that shouldn't be in the brain normally or in the body normally. So there's a number of approaches that are now underway, which is to see whether we could utilize the body's own innate ability to degrade and target these foreign bodies or these pathological species of alpha-synuclein in order to degrade them. And there's really two main approaches that are being done to try to do this, and both of these are now being tested in the clinic. In one case, what some teams are doing, which is to use in a laboratory to genetically and generate antibodies in the laboratory that will target the pathological forms of alpha-synuclein, these protein clumps. So that is to develop in a laboratory a very specific antibody that will target the pathological form of alpha-synuclein, these clumps, and then inject that antibody into somebody with Parkinson's disease to see whether you can now target that antibody to the brain and have that antibody induce the body to degrade the protein clumps. So that is one approach, and that is actually, there's two different companies now that have gotten to clinical testing with their laboratory-generated antibodies for alpha-synuclein. The other approach that a different company is now in the clinic testing is more based on sort of a more traditional type of vaccine approach. In this case, they've generated in the laboratory a fake version of the protein clumps. And what they do then is inject that fake version into people in order to induce an immune response. So the body then, in response to getting exposed to this synthetic version of the protein clumps, generates an immune response. The body itself makes antibodies and activates its immune system. And the concept then is that that activated immune system will be able to go into the brain and degrade the protein clumps seen in disease. So those are the two approaches that are going. Sorry, go ahead.
No, that's okay. I was just wanting to get a sense of where we are now, and perhaps you can give us a sense of that, Todd, about what we're learning so far. These are fairly far along in the testing process, right?
That's right. All the approaches I just said, whether they're the laboratory generated antibodies or this more vaccine type of approach, have all reached clinical testing. In the cases of the antibodies, they have tested them in people without Parkinson's at first to look at safety issues and also to see whether they can alter some of the measurements levels of alpha-synuclein. And both of those studies hit those endpoints, so they were seen to be safe and having an intended biological response. So they're now moving forward towards testing in Parkinson's patients. The vaccine approach has been tested in a small number of Parkinson's patients. It has been demonstrated that, in fact, an immune response against alpha-synuclein is generated when someone is exposed to the synthetic form of the antibodies. So now that is also moving forward into further testing to try to evaluate the consistency and prolonged duration of response, and ultimately we'll be moving towards trials to look at whether we can impact the symptoms and progression of Parkinson's with these approaches.
And Dr. Kalia, one thing that Todd referenced that I think is reason for optimism and encouragement in this area is that there has been some success in the Alzheimer's field with a similar approach, with an antibody approach to fight against the protein at work in Alzheimer's, different sticky protein, beta amyloid. But it sort of gives a kind of proof of concept, right, that if that may be working, hopefully in the Alzheimer's arena, that a similar approach could work in Parkinson's. Is that right?
Yeah, I think so. I think previously there were beta amyloid, so is a protein, one of the culprit proteins in Alzheimer's disease, whereas alpha-synuclein is the main culprit in Parkinson's disease. And I think earlier our thoughts were that maybe they were quite different because beta amyloid is found outside of the cells, which would then make it quite amenable to being recognized by these antibodies. And the original thinking was that alpha-synuclein stays inside cells. But there's now a lot of evidence, actually, that alpha-synuclein, although it's found in cells, can actually leave a cell. And in many circumstances, it's the alpha-synuclein outside of the cell that is the one that we really want to target, because that alpha-synuclein can move on to a neighboring cell and can cause that cell, the alpha-synuclein within that cell, to take on these clumps. So I think the evidence from the Alzheimer's field showing some good, they've done a fair amount of work, and there's been many years of investigation into this, but there's a lot of promise for the antibody therapy against beta amyloid, and I think that does portend, hopefully, a very good prognosis for alpha-synuclein as a target for immunized-based therapies.
And let's take a look now at one other way of fighting against alpha-synuclein. In addition to this idea of the immune system going to work on our behalf, there's also the idea, Chair, of actually preventing the clumps in the first place. So this is sort of an alternative approach, which maybe you could comment on that as well. Part of what's exciting about all this is that we're not just putting all our eggs in one basket, but we have a variety of strategies. And this one would try to get it sort of more at the head end, right, to prevent the clumping in the first place?
Yeah, so this other approach is actually using a more traditional small molecule-based approach. So this is more the typical type of therapy that we're used to, which is think about a pill-based type of therapy. And this particular compound is designed to keep everyone has alpha-synuclein in their body, whether you have Parkinson's disease or not. And what we're trying to do is keep alpha-synuclein more in its healthy, normal structure, which would be more to the left on this slide, and prevent it from getting into these clumps and more the pathological form, which is on the right, the fibrils and the Lewy bodies. So this compound is designed to allow alpha-synuclein to maintain its normal function that we all need for it to be performing in the brain, but prevent it from getting into more the pathological state, which are the Lewy body clumps. This compound has also entered clinical testing, at first a safety study in people without Parkinson's, and is now moving forward based on those results as well.
And before we get to our next approach, which are this idea of these compounds that may either reduce risk or slow down disease progression, we've got a number of questions coming in. And so let me take up one of them right now, which is a good question that came in from someone about the alpha-synuclein trials. And that is they want to know, Tashir, whether or not there have been any adverse side effects so far from these early testings. You were saying that so far the safety has looked good in these tests, but any problems to date with these early trials?
Yeah, so far there has not been anything significant that has come out that would prevent the projects from moving forward into additional testing. I think it's a really important question, though, because as I had mentioned, alpha-synuclein has a normal function in the body, in everybody. And it's expressed not only in the brain, but it's expressed in blood and a number of other tissues. So there's a lot of effort being put into really dissecting these early trials to make sure that we are have the potential to have a therapeutic benefit and targeting the pathological forms of of alpha-synuclein, or the forms associated with the disease, and not really having a dramatic or any impact on the normal function of alpha-synuclein. And so far that seems to be the case. These have been relatively small studies, so there'll be further testing to dissect this out further.
And Todd, one other quick question before we get to our next approach, which is the sort of $64,000 question in all of this, which is one of our participants wants to know how soon this will be available. Of course, there's still a ways to go with more testing and then the regulatory approvals, but if all goes well, let me put it, I guess, that way, when might some of these alpha-synuclein products, or alpha-synuclein fighting products, rather, be potentially available to people with Parkinson's?
Yeah, I mean, this is still, in terms of clinical testing, relatively early. It's exciting, of course, that it's made it into clinical testing. One of the challenges, and we'll talk about this a little bit later in the conversation today, is, the goal of these treatments are to see if we could slow the progression of the disease. So unfortunately, those studies, those clinical trials, do take some time because we want to see, is someone better off, 1 or 2 years afterwards after taking this treatment. So that's hard to speed up. So, I mean, if all goes well, I think we are still in these areas probably 5 years or so away. But that would be, really hoping that all goes well with these particular therapies.
Okay, thank you. Let's move on to another approach, and we'll come back and take more questions about alpha-synuclein as you have them. But we want to get to this other fascinating area that Dr. Kalia was describing earlier, which are these different products, different compounds that seem to have this associational effect, where we see people who take a particular blood pressure medication or some other compound, and they're less likely to get Parkinson's, or their Parkinson's progresses more slowly. So the first one we want to explore, and we want to bring Bill Sheppard into our conversation here because he actually participated in this trial. But this has to do with a particular blood pressure medication that's been tested, a drug called isradipine. And this is a drug that's used for people with high blood pressure to sort of block the calcium channel. And before we get Bill to comment on his experience in participating in this trial, Dr. Kalia, maybe you can explain why it is that something that would work perhaps to lower someone's blood pressure might be effective in Parkinson's, because it has to do with calcium, and we think that calcium has something to do with the degenerative process that happens in Parkinson's. But give us a little bit of a 101 on this, and then I want Bill to describe his experience in participating in the trial.
Okay. So in cells, in the cell wall, there are things that we call channels, and they allow particular ions or chemicals to cross the membrane of a cell. So blood vessels, cell wall, blood vessel cells have these, heart muscle cells have these, and brain cells have these. And it just so happens that a particular blood pressure medication was developed to be able to block a channel that allows for calcium to enter a cell, and its initial indication was really to lower blood pressure. Work in the lab, though, had been going on that demonstrated that brain cells also contain this calcium channel, and in particular cells that are very vulnerable to cell death in Parkinson's disease, for example, the substantia nigra neurons contain this calcium channel. And via a variety of different mechanisms, calcium can be quite harmful to a cell. So if you have too much calcium, that actually can be a signal for a cell to die. And so the hypothesis was that if you block calcium, these calcium channels, prevent calcium from entering these brain cells, that perhaps this could be protective for the cell. And indeed, a lot of experimental evidence has supported this. And interestingly, in parallel to this experimental evidence, people looked back to look at populations of patients with Parkinson's disease to see if there was any hints that maybe this was occurring in patients as well. And surely enough, patients who were taking these kinds of drugs that could block this particular calcium channel seemed to have at least an association. Like I mentioned, we can't definitely conclude causality, but there was definitely an association that if you were taking one of these types of blood pressure pills that blocked this particular calcium channel, that it appeared that your risk of developing Parkinson's disease was less. And so that was a very exciting finding that had led to this initial study PD trial that Bill Sheppard was involved in.
So Bill, let's hear more about your particular story. This is now fairly far along in testing. You actually participated in some of the early initial safety testing, the phase 1 portion. Describe, what brought you to wanting to participate in this particular trial, and then something about what your experience has been like.
Yeah, so I read about some of this research that was mentioned back in 2005, 2006, or the year after. It was done at Northwestern. I was living in Chicago at the time, and they announced they were looking for participants in a phase 1 trial. It looked like a very promising drug given the epidemiological and science-based evidence that existed. It felt like something that was very low risk because isradipine had been around as a blood pressure medication for several decades. And sure enough, when I called in to Northwestern, they did have openings in the study. It was about I forget, 10 to 12 week study, basically ramping up on isradipine and ramping back down. It was phase 1, so I knew what I was taking. It wasn't a double-blind study. And it was pretty straightforward. You were checking your blood pressure a couple times a day. You were seeing the doctor every couple of weeks for the duration of the study. And then when the study finished, I asked the question. I said, how many people are how long until the study is going to be complete and you have results? And they said, well, probably 6 to 12 months, but it depends on how quickly we can recruit the remaining number of people we need for the study. And I said, how many people are you looking for? And the answer came back with something like 16. So here we have this kind of great drug, that had a lot of promise, very low risk characteristics, and we were holding up progress on the fundamental science because we couldn't get 16 people into the study, which was really a wake-up moment to me as to how important it is for the Parkinson's community to help drive clinical trials and the science forward by actively participating in them wherever possible. Because research has slowed significantly by the timeline to recruit patients across all of these studies.
You'll say a little bit more about that and what you now say to patient groups when you're with our fellow citizens who are experiencing this about making the case, because this is a pretty crucial point, as you indicate. As I understand it, something like only about 10% of those of us who have a Parkinson's diagnosis actually participate in clinical trials. What do you say to your fellow people living with Parkinson's to make the case about why this is so crucial?
Yeah, I'd say, there's a very wide range of trials. I've probably participated over time in 5 or 6 different ones. A lot of them are just observational, giving blood samples, doing a DNA test, doing something in an MRI machine, are all things that I've done over time. And the commitments vary. It can be as short as a half hour, and it can be as long as a multi-week or multi-year commitment that you make. But generally speaking, these are zero cost to you. In fact, you get reimbursed for travel costs, in my case. There is a tremendous focus on safety by everyone involved in the trial, and patient safety is first and foremost in people's minds. So I felt like I was very well taken care of from that point of view through the trial. And there's very fulsome disclosure. And that's part of, as you would expect in today's society in the U.S., there's extensive disclosure on risks, and the doctors who are part of the study will spend generally as much time as they need to with you to make sure you fully understand that side. The pluses, you get seen by a world-class clinician, stay on top of the research that's going on and learn more about the disease that's affecting all of us or our loved ones. And oftentimes you're on the leading edge of being able to benefit from an improved treatment. Back then it was very hard to find out about studies. You had to do what I do, which is run across stuff on the web and do your own homework. Now we have the Fox Trial Finder, which is a fantastic way for people to find out about very easily clinical trials in their area that may be relevant for them. And we all benefit from the research that went on before us in terms of the drugs that are available to us as a Parkinson's community today. Participating in a research study, whether you have Parkinson's or not, because they're always looking for controls, is a fantastic way to both give back to those who came before us, help ourselves, because many of us will be hopefully around long enough to benefit from continued advancements in treatment, and hopefully benefit future generations. Not everyone can write a huge check to fund research, but just about everyone can participate in clinical trials and help drive the research forward. I'm a huge advocate for doing so.
And Bill, a number of people are writing in wanting to know the next kind of obvious question here, which is, again, you can't run a controlled experiment on yourself, but what's your own? People want to know what your own sense is. Do you have a sense of whether or not participating in this, taking this Ratapine, was useful to you in your Parkinson's?
Yeah, I mean, I've been blessed with a very slow progression of the disease, but it has progressed over the last decade. I have continued to be on Ratapine continually since then, based on conversations with my doctor. But I think it's important to note that I'm a statistical sample of 1 and therefore invalid. So I wouldn't read anything from my experience other than to note that the drug has continued to progress through the process, and with the phase 3 trial going on, hopefully we'll all have more definitive answers soon as to whether or not Ratapine might be neuroprotective.
Well put, I think, because it is true. It's so tempting to want to generalize from, as you say, a sample size of 1, and we just don't know definitively that answer yet. But it's also, I think, a cautionary tale about how long this takes, given that you started this process in 2006 or 7, it sounds like, and here we are 8 years later, and we're still awaiting a definitive answer. let's, Bill, stay with us, and we'll involve you as our conversation as we go forward. But I want to ask Dr. Kalia one question about this that also has come up, which is one thing I think we would have to be cautious about with isradipine, and why probably people shouldn't just run out and start taking it, is that many people with Parkinson's, I'll offer my own sample size of 1 here in my own case, I have really low blood pressure, and which is quite common in Parkinson's disease. So we have to be cautious about this too. It's not like everyone can just rush out and start taking this drug because of that other concern.
Yes, definitely. So first of all, I think it's important from the point of view of balancing risk and benefit. Right now, we don't know what the benefit of isradipine is, and I think that going through the process of having a well-studied medication is important before we decide whether or not to pursue treatment. And secondly, yes, one of the potential side effects of isradipine, because it is a antihypertensive, is that it could reduce your blood pressure, and if you're doing this on your own without actually being medically overseen, you could run into problems, and particularly because Parkinson's disease can be associated with a lower blood pressure, and some of the medications that we give for Parkinson's disease can drive that blood pressure lower. So this is not to encourage people to take isradipine, but I think one of the optimistic findings from this initial safety study that Bill was involved with is that the overall, from an adverse events point of view, there was some dizziness, but this didn't appear to be associated with low blood pressure. So I think, although that is a potential side effect, I think that it may be less prominent than we see with some of the more aggressive antihypertensive drugs. But yes, I completely agree with the message that people shouldn't be rushing out and purchasing isradipine and taking it on their own, and that we should really, if somebody is interested in that, they should really maybe try and find a center where they could participate in a clinical trial, and then, as Bill already described, really contribute to the research and really get a solid answer on whether or not this is going to be a medication that we could take forward for neuroprotection in Parkinson's disease.
Yeah, it's such a key message because, well, it's frustrating to wait so long for some of these results. The only way we're going to speed the process is, as Bill suggests, all of us participate in research. Let's move on and take a look at another one of these compounds that has been associated, at least, with a lowering of disease risk, and that has to do with a substance called urate. It appeared on our screen for a moment, but it disappeared again. So let's see if we can get it back on the screen and see what that involves. There it is. So Dr. Kalia, while we have you with us on this slide, why don't you pick up on this one? This is another interesting observational study, and again, we're sorry, our bullet points keep disappearing. But go ahead, if you would, Dr. Kalia, and describe what urate is and what it is we're trying to find out about it in terms of its lowering risk capabilities with Parkinson's.
Yeah, so urate in and of itself, or uric acid, at least in studies that we do in a test tube or a cell culture system, or even in animal studies, it appears to have antioxidant capabilities. So we know that free radicals and oxidative species can be harmful to cells, particularly brain cells, and if we have ways to reduce this with antioxidants, that can be protective for cells. So a fair amount, again, similar to the isradipine story with calcium channel blocking, a fair amount of experimental evidence has suggested that if you can actually add urate to conditions where there is increased cell death, you can protect cells from the process. And similarly, again, with the epidemiological studies, that if you look at populations of patients with Parkinson's disease and you measure in their blood the amount of urate they have, that patients who have higher levels of urate seem to have a lower risk of developing Parkinson's disease. And also, interestingly, if you have Parkinson's disease and you have a higher serum urate level, that your progression of disease seems to be slower. So this was encouraging, and it had obviously led to this clinical trial that we're going to talk about. similar, again, I think to his Radipine story, is that it appears to be quite safe in these initial studies, because there are concerns. If your urate is too high, you can develop things like gout. You can have some cardiovascular complications. You can develop some stones in your kidney, so renal stones. And at least the initial safety trials don't demonstrate that these side effects are going to prevent us from going forward with further trials. So that's the story for urate for now. So I guess so that's urate itself. What the actual drug is that's being tested is inosine, which is a precursor to urate. So what the actual intervention is not to give patients urate by mouth, but you give them inosine, which within the body gets converted to urate.
And this one, both of these, we should note again, are fairly far along. They're in that phase 3, that last step before regulatory approval. And so while it's been a long road, as Bill Sheppard was describing, in this instance, both with isradipine and with inosine, we should have a definitive answer, we would hope, within a few years. Let's look at one more example, and this is in some ways particularly fascinating because it shows a relationship between something we have traditionally thought of as not being good at all, nicotine, and its potential effect on slowing down Parkinson's disease. Todd, share, if you would please, just a brief description of what we've learned so far and what we still hope to learn.
So the main driving piece of evidence that led to exploring nicotine for disease modification in Parkinson's came from a number of epidemiological studies that suggested that smoking was actually associated with a decreased risk of Parkinson's, and that's for even controlling for sort of the obvious explanation of life expectancy. So controlling for life expectancy, people that smoked seem to have less chances of getting Parkinson's. So just with the theme of what's been discussed today, that was just a correlation, and in fact, within smoking and cigarettes, there's many chemicals. But of course, the attention quickly went to nicotine for another reason, in that nicotine actually functions by interacting with specific receptors or proteins in the brain that function in the motor system. So there's sort of that extra piece of evidence where it maybe makes sense because nicotine is targeting the same brain region that's impacted in Parkinson's. So this led then to a study that's been ongoing, and we should be getting results probably within the next year, which was to explore whether giving Parkinson's patients early in the disease nicotine, in the case here they did it through a nicotine patch, could actually have an impact on the progression. So could we convert that correlation, that link that was seen in the epidemiology with actual treatment opportunity? So that study is ongoing. I think it goes without saying that there are a lot of side effects of smoking, so we wouldn't recommend anyone start picking up smoking. But this study focused on the patches, I think, will be pretty interesting to finally really be able to determine whether this correlation with smoking has an effect and can be converted into a potential treatment.
Thank you. I'm going to push us along here because we still have some additional information we want to get through, and we want to be sure to have room for some of your questions at the end of our hour. So we're going to spend a brief moment on something called neurotrophic factors. This is something where there's been a lot of excitement over the years about the potential for these so-called growth factors that are actually these proteins that help support dopamine cells in the brain, nurture them. Some people compare it to like fertilizer for the brain. It helps those neurons get stronger and function well. And it's been very difficult to convert that, though, into something that has actually worked well in actual clinical trials. So, and so we're going to just go right ahead and look at what some of those problems have been, because part of what happens with neurotrophic factors is that you can't just take a pill. It doesn't cross the blood-brain barrier, and therefore it requires a surgery, and that's where it gets tricky because you need to get it into the right place. You want to make sure you're just growing the neurons and not other things in the brain. It's been a very tricky process, Lorraine Kalia, and yet people haven't quite given up on it yet. As I say, this has been an area of intense interest and excitement and also great disappointment. So if you would, Dr. Kalia, just describe briefly what our hopes still are, despite all of the kind of roller coaster ride that neurotrophic factors have been on for many years.
Yeah. So I think there's been a lot of study and there's been a lot of hope in this area. And although there have been a number of kind of disappointments, I think that this is an area that warrants still some optimism. And perhaps one of the biggest challenges, which you've already alluded to, is challenges of delivery. It was quite evident early on that to be able to use this therapy, we were going to have to get into the brain somehow. In some cases, there was injecting of the neurotrophic factor just into the ventricles, so the fluid-filled spaces in the brain, and hoping that it would reach the right regions. That didn't appear to be feasible. The second approach was then, okay, well, if we can't just kind of put it in the brain and hope that it finds its way to the right place, then maybe we need to inject it directly into those areas. And so one of the more recently studied growth factors, neuturin, was initially injected into the striatum, which is a portion of the brain where the substantia nigra neurons communicate with and project to. That, again, didn't seem to be sufficient, and so then the last trial that was done was injecting viruses that produce this growth factor in the striatum as well as the substantia nigra. And again, unfortunately, the outcomes were not as we had hoped. Patients who received the treatment or patients who didn't receive the treatment didn't show any differences. But still, I think part of the thought within particularly the surgical community is there's still probably things to be learned from delivery. There are newer techniques, convection-based techniques, which allow for more distribution within those brain structures, because even with injecting into these two different brain structures, there probably wasn't enough spread of the virus within those areas to cover the area sufficiently. And I think the other take-home message from these trials, at least, is even if it turns out that growth factors aren't going to be the answers for Parkinson's disease, this area of investigation has really at least allowed us to develop and to study the use of what we call viral vectors, which are viruses that express proteins in the brain, and that at least in this context have shown them to be effective in terms of being able to express a protein, but also safe. And so even if what we learn from this whole area of study is not growth factors, it may actually be able to be translated over to another area of investigation in terms of expressing other proteins that may be important for being able to give neuroprotection. So I think, although there have been some disappointments in this area of investigation, I still think that there have been some excellent learning points from it.
It's a really interesting point. So even though the content may not be right in terms of growth factors, the technique of using these viral vectors to enter into the brain and express a protein that could provide benefit, that technique may yield benefit with a different substance later on. We also want to draw your attention to the last bullet point, which is that there is a way to sort of do this in-house to some degree, and that's through exercise. Exercise has been shown to boost neurotrophic factors, and as many neurologists will now say, and I think you probably concur with this, Dr. Kalia, that exercise is something that can be of great benefit, and has the benefit, of course, also of not involving brain surgery. So it's something that people can do.
Definitely.
Let's move on to our last point, and then we can get to more of your questions. And Toucher, this has to do with the challenges involved in testing any of these. And it's, the first point is the one that Bill Sheppard made so eloquently, that we need people to participate. We're not going to be able to test these various disease-modifying therapies unless people participate. But beyond that, there's also the question of having the right measurement tool. And this is something that the Fox Foundation has put an awful lot of effort into. But if you would, Todd, just describe sort of where we are in developing a way to actually test whether or not these various techniques that we've been talking about actually work?
Yeah, so I think one of the most important things that we're working on in parallel to developing the specific therapies is to try to figure out ways to monitor and assess their effectiveness earlier in the process. So as I mentioned earlier on the call, the goal of these therapies is to try to slow the disease. And right now, in order to see if someone, again, is better off 2, 3, 4 years after taking the medicine than people who didn't take this potential medicine, there's no way of doing that study without that number of years. So if you want to see, are you better off 4 years later, that takes 4 years. So if you want to see if you're better 6 years later, that takes 6 years. So we need to figure out a way to do that more quickly and get an indication of an effect. And that's where the biomarkers come into play, where you could get an insight earlier in the process of whether you're impacting the disease. Just as an example to give people would be a measure like cholesterol, which can tell you whether a medicine is decreasing your risk of heart disease without having to actually wait to see if somebody gets heart disease. You could just get that blood test and see very quickly whether the medication has had an effect that you want. So we're looking for something like that for Parkinson's. And there's been a pretty significant effort looking at blood-based markers, brain imaging technology, measurements in the cerebral spinal fluid, which is sort of a surrogate for what might be happening in the brain, as well as trying to develop improved clinical measures, more objective measures of clinical symptoms, motor function, that can give us this better insight into the disease progression and ways that we may be impacting that progression. And there's been a lot of progress in this area in terms of new imaging technology. And, for example, we can now measure alpha-synuclein levels in spinal fluid as a potential indicator of disease. So this is an area of incredible importance because any breakthrough in the biomarker space would dramatically accelerate our ability to test any of the therapies we mentioned and any that might be coming down the pipe.
And it's such a key area, again, for people to be able to participate in the various biomarker studies that are going on, because we're not going to know if we have the right tool to modify or slow disease progression unless we have a way, as you suggest, to actually conduct measurement in an accurate way. And it's why, of course, both participating in that kind of research and investing in that kind of research is in many ways is as crucial as investing in the actual research to find a disease-modifying entity itself. Let's get to all of our questions now. I'm going to jump us to our last slide because a lot of people are wanting to know how people can participate. And Bill Sheppard, come back in on this question if you would. You mentioned foxtrialfinder.org, and a number of people are asking about how they can find studies, how you figure out which study they could participate in, because sometimes you have the opportunity to participate in many studies. So give us your advice, Bill, about how you go about selecting a study to participate in, because some are more demanding than others. What's your advice to people who want to participate but just aren't quite sure how to go about it or what it might involve?
Well, I should probably let Todd speak to it, but I think in response to trying to drive research forward faster, the Fox Trial Finder tool was created to give people a place to go to find the studies that are out there on Parkinson's disease. Todd, I don't know if you want to.
Yeah, so the goal of the website is to give a one place where you could search and learn about every trial that's happening, and you can search by geography or by particular topic you're interested in, by stage of disease, and it allows you to then connect directly with the clinical centers to learn more about what's happening with those trials. So it's really more aggregating all that information. As Bill mentioned, when he got involved in isradipine, he had to do a lot of sort of one-off research on his own. So this compiles a lot of the information in one location to make that easier.
And the tool also lets you enter the characteristics of where you are in the disease, because a lot of times the trials are looking for people who are newly diagnosed. I'll come back to that in a second. They're looking for people who have been on, who have certain symptoms, who are having dyskinesias, for example, or They're looking for people who have not yet started Met or levodopa as a treatment therapy. So by putting in your characteristics, you can get a regular email about trials that are in your area that fit the characteristics on a preliminary basis. I would encourage people to participate in as much research as they have the time and energy to do so. I think it all helps drive toward an end goal of finding a cure or set of cures for Parkinson's. Go ahead.
And I guess the other point about that I think is key is that it gives you a menu, it gives you a choice, so that if you want to participate in something that's perhaps less time demanding, it gives you those possibilities as well. It's not like every trial that you participate in is necessarily intense, either in terms of time or even the kind of drugs that you might take. Some are just observational studies. There are a lot of different possibilities, and it's important to keep that in mind as well. Let's get to the rest of our questions in the time we have remaining that have come up, because there are a lot of interesting ones. And one of the ones that's come up that we haven't talked about yet and that people want to know about, Lorraine Kalia, are stem cells. So much hope, not unlike trophic factors in a way, for what stem cells might offer. Been a lot of obstacles, not unlike trophic factors, kinds of obstacles but where are we now, and whether or not stem cells might also provide a way of slowing down disease progression?
Yeah, so I agree that there are some parallels with the nerve growth factor story, in that there's been many years of investment into it. It's a much more complicated kind of treatment because, similar to the neurotrophic factors, it also requires a neurosurgical intervention. You can't just get stem cells into your brain without doing some sort of surgery. I think one of the important points that we've probably learned, and I think is probably pretty accepted in the field, is that stem cells in and of themselves probably aren't going to be the full answer from the point of view of neuroprotection. The way that stem cells were looked at in the past were to take stem cells, implant them into the areas of the brain where you'd hope that there'd be some regeneration, so into the striatum, for example. And the whole goal was basically to hopefully have cells that would produce dopamine and, in a way, be a very localized dopamine delivery system. It turned out to not be as straightforward, and similar to the neurotrophic factor story, there are a lot of technical issues involved in terms of how those cells are delivered, how much tissue coverage you get, how well those cells survive, and making sure that those cells don't develop into something you don't want them to. And similarly, because you don't have the control of the dopamine delivery from these cells, that some people developed a lot of side effects, such as significant dyskinesias. I'd say probably a couple of years ago, people probably would have said that the whole stem cell story was dead. But I think reevaluating the studies now that we've had a fair amount of time, and I think this also goes back to the messages that we were talking about earlier in terms of how much can be learned over time. I think there's some resurgence in the idea of doing stem cells. There's a study going on again in Europe. But in addition to that, technologies have changed. So in the past, you had to be able to get stem cells from embryos. Now there's a huge amount of research, an exciting area of research where you can actually take cells that aren't brain cells and convert them into brain cells, and even more still can convert them into dopamine-producing brain cells. And so although still very, very much in its infancy, because this definitely hasn't gone to humans yet and is being studied in cell culture systems and in animal models, this may be a different avenue by which to go. But whether or not that's going to be the answer for a disease that we know that affects the entire, many, many regions of the brain, probably is not going to be the full answer for neuroprotection, but maybe a useful tool to be able to allow for dopamine delivery, or at least some regeneration of cells within a specific area.
Thanks for that summary. And again, it's a point, I think, important point for all of us to realize that we really need a pretty broad arsenal of possibilities, because it's not likely that there's going to be a single bullet that's going to take care of the many faceted aspects of Parkinson's disease. Todd Scherer, there are a number of people writing in who want to know whether or not any of these ideas that might slow down disease progression, let's go back, for example, to the alpha-synuclein question, would actually also improve you symptomatically. In other words, it's one thing to sort of stop disease progression, but would it just hold you at that point, or would you actually improve?
Yeah, I mean, this is actually an important question, and it's a little bit of a philosophical question also. In terms of the alpha-synuclein-based therapies, we don't know the answer to that. So you could have a hypothesis that the clumps within the cell actually are preventing sort of the communication within that brain cell. Think of it as like a traffic jam within a brain cell. So if you remove that traffic jam, all of a sudden you can get through the Lincoln Tunnel and get into Manhattan. So you'd get sort of an immediate benefit, but then sort of the tunnel remains clear. So over time, you're also progressing. There's not a traffic backup, so you're not progressing sort of at the same rate either. So that is going to have to be determined in the clinical trials, and the researchers will be looking for both an early symptomatic benefit as well as the effect on progression. Ultimately, we are only really interested in the symptoms of Parkinson's in terms of these clinical trials. So whether you slow the progression of a symptom or have an immediate symptomatic benefit, that is really the goal. Like, for example, the biomarkers are just going to be tools. We don't particularly care, for example, that we lower someone's cholesterol. We care that they don't get heart disease. So I think that is, ultimately the goal. If somebody's symptoms progress more slowly and/or they have an immediate symptomatic benefit.
And Todd, some optimism there again from the Alzheimer's field, in that the early studies in one of the immunotherapy approaches to Alzheimer's not only showed a lessening of the sticky protein beta amyloid, but it also indicated some improvement in symptomatically, right? So that's also an encouraging, at least, possibility.
Yeah, so in Alzheimer's, at least in the small study that we're talking about, they showed a cognitive improvement that matched with the removal of the aggregates in the brain.
So a lot of questions, Lorraine Kalia, about calcium, since we brought that up, and that was the trial that Bill Sheppard participated in with his isradipine. People want to know, I think, logically, whether or not they should then add calcium to their diet, sort of like what you do, if you're worried about osteoporosis. Or should they not do that? Should they not drink milk or eat yogurt? And it's one of these things that, people make an association almost like, well, gee, if the problem is a sticky protein, should I reduce protein in my diet? Give us your sense of that question.
Yeah, so it's a logical thought, but it's a little bit more complicated than just calcium levels. So it's not calcium levels in the blood that is related to what this observation was. It was the use of this particular type of drug. And so most definitely people shouldn't alter their dietary calcium based on what we discussed today. They should take the calcium that's been recommended either from their food guidelines, or if they, for example, have osteopenia, or if their family doctors have recommended that they be on a calcium supplement, then they definitely should follow those instructions. And it's not actual calcium levels in the blood that are the concern. It's these particular calcium channels and the way that they function on the cells. So yes, definitely no changes in your calcium or your diet related to this.
Bill Sheppard, a number of people have written in saying that they're kind of overwhelmed by all the choices that are available to participate in trials, and wondering if you have any advice about how to go about selecting which clinical trial or observational trial to participate in.
Yeah, I mean, I think you have to look at which ones you qualify for, and what do you have the time and energy for, because you can't do them all. But I think the ones that are reasonably nearby you, and if you're near a major city with a major research center, Chicago and New York and LA, you're probably going to have more choices than if you're in the middle of Wyoming or something. I think I'm not sure every trial is a good trial, but there's probably good in every trial. If you've got the time and energy, I think participating in any of them can be worthwhile. And the ones that I think are most important ultimately are the ones that are help with things that are neuroprotective or neurorestorative. Those are the things we're trying to get to as an endpoint. But a lot of these things do a better job of treating symptoms, and for all of us who live with the disease, we know how important that is to our day-to-day life. So I wouldn't necessarily discriminate. And advancing some of the core science in these observational studies is also very important because it creates the roadmap that leads to creation down the line of additional drugs or treatments.
Thank you, Bill, and thanks so much for your participation in our conversation today. It really helps to get that sort of first-person viewpoint on both what the experience is like of participating in a trial, but also underlining the importance of doing just that. So,
thanks, Dave.
Thank you.
Thank you, Dave. And I wanted to just add one last thing: is if you're open about the disease with friends and family and or colleagues, which I've been over the last 10 years. I find it's not uncommon that somebody who just gets diagnosed gets in touch with you to talk about it, connected by somebody. I'm sure a lot of you with Parkinson's have experienced that. Those folks are particularly valuable to the clinicians because a lot of times clinical trials are looking for people who are early on in their diagnosis stage. So encourage everyone as they run across other people with Parkinson's, and especially the newly diagnosed, also encourage them to look at the research that's out there and try to participate.
A great point. I think all of us have had those conversations with people who have gotten in touch with us, and it's a great point to remind people of that opportunity, especially early in the disease course. Really want to thank you, Bill, for all you've done and for your participation in our conversation today, along with thanking Dr. Lorraine Kalia and Dr. Todd Scher. Thank you both. Thanks all three very much for participating. And thanks to all of you for joining us on this webinar. As always, we had lots of great questions. As always, we did not get to all of them, though we did get to many. And we do want to remind you that you have an opportunity to take up your questions further tomorrow when we do our Twitter chat on this topic. The hashtag for that is FoxPDChat. That's tomorrow at 12 noon on Eastern. 12 Eastern. So thanks again for everyone for participating in our webinar today. We'll be sending you a link so that you can listen again on demand and also fill out a survey form which helps us learn what we can do to continue to do our best to improve these Third Thursday webinars in the future. Join us again next month on the third Thursday when we are going to take up in November the question of balance and gait issues, something that have also been a difficult problem to solve, and we're going to give you the latest on what we know about solving the problems of balance and gait in our third Thursday webinar in November. Thanks again, everyone, for joining us. I'm Dave Iverson.